96. It's just so many to draw and to understand what it looks like when there's 92 and when they line up and when they split apart. So if you could give yourself some sort of practical number and try it. So you could try writing it how I write it, but then try it with a different number of chromosomes and make sure that it makes sense to you. Because sometimes it's one of those things where it looks like it makes sense, and then you go to write it and you're like, wait, is that right? And so I think a lot of students just get confused with chromosome number. So the interphase and mitosis and cytokinesis are the three main blocks of the cell cycle. So interphase is G1, S, and G2. So G1 is where the organelles will replicate. So it's a good idea to identify, see if you can name 12 organelles. We can go through those together also and name the function of the organelles. And then replication happens in interphase. So remember the primary polymerase is DNA pol delta. It looks like a S. S reminds me of synthesis, so that's how I remember it. And then, so we're here in the nucleus, and then the nucleus starts dissolving, and they start lining up and all that kind of stuff, which I don't really care that you know, like this is prophase, this is metaphase, and it's for I think that's for another class. Our whole point is, I like to talk about chromosome number, especially that we know the difference between mitosis and meiosis when we talk about sexually reproducing organisms, and what the point of sexual reproduction is genetic variation, and genetic variation leads to antibiotic resistance in those organisms, or fungal resistance to fungal medications. And so anytime you add genetic variation, they're more likely to be able to mutate against whatever pharmaceutical that patient is taking. So I think that part's important. Okay, so in mitosis, there's PMAT, prophase, metaphase, anaphase, telophase. The one we really care about is metaphase. Basically, it's all coming down, what this whole system is. The whole system is nucleus dissolves, or the chromosomes line up, the chromosomes split, and then the chromosomes are pulled apart to their separate poles. So as long as you kind of understand what the whole point is. And then when they line up on metaphase plates, so here they're in the nucleus, we went from 4 here to 8 here, and then the 8 line up on the metaphase plate. And then when it goes through telophase and cytokinesis, they've separated, the nucleus reforms around the chromosomes, and we end up with 2 cells that have undergone the whole process of cell division, and they're genetically identical, hypothetically. Yeah.
I've always been curious about this. So in the process of like protein synthesis,
yeah.
Does it stop when the cell is going through replication? Is it only happening during G1 phase of the cell cycle?
During G1 phase of the cell cycle is where all the organelles will replicate. But protein synthesis, it's different for all eukaryotes, I would say, for each different eukaryote. But this whole thing will occur, and these cells will separate, and then these two will go about the business of protein synthesis separately when they're fully formed and shaped. Because basically this is like childbirth, like cell birth. Yeah, so once it goes
like, is it paused when it's going through replication?
That is a good question. I would say yes, because what's happening is the origins of replication are what is euchromatin, and the rest would be heterochromatin. So the euchromatin areas are the origins of replication. The origins of replication are unsupercoiled at the point, and then everything else would be supercoiled. So that's my guess. But maybe there are cells that can do it simultaneously. Yeah, I'm guessing it's separate, but that is a really good question. Yeah.
This is asexual reproduction for single-celled organisms?
Yes, so that's a really good point to bring up, because don't forget, this is one cell of one something. But, so if you talk about like a yeast, we're going to talk about what the difference is between yeast and molds today. So if this is one cell like a yeast, that this cell lives on its own, functions on its own, does its own stuff, will replicate. Now there's two yeast cells that are genetically identical, but this one will go out and live its life, this
But
one will go live
two
its life.
separate organisms.
As two totally separate organisms.
Okay.
That came from one organism. So that's like a single cell. But if you talk about like a penicillium mold, there's yeasts and then there's molds. And if you like zoom into one of these little areas, this is a cell, this is a cell, this is a cell, and these are individual cells, and this one cell can replicate and add another cell here, but it's part of the bigger whole, but it goes through the exact same process. It'll just
Okay.
go through interphase mitosis and cytokinesis and separate into another cell.
Either way, it's called mitosis.
Yes,
Okay.
yeah, yeah. So either way, they're going through the cell cycle. Mitosis is part of the whole cell cycle. Does that make sense? Yeah, how that works? I know the single cell organism versus a multicellular organism trips everyone up. Like everyone is like, wait a minute, that it's confusing. But you know, where to add another level of confusion, where yeasts are and molds are funky is that this cell is part of this organism. And this cell acts as an individual organism, but this cell can split off and go form its whole own mold organism. So they kind of can do both. They kind of can sexually reproduce and asexually reproduce, and they can.
And
it
this is
can
mold?
be single cellular. It can be multicellular. So
Mold can sexually reproduce and asexually?
sexually and asexually reproduce. Yeah.
Fabulous.
So can yeast. Yeah. So it's weird. It's just like once you get to fungi, you're like, wait a minute, because they can do either or. So they can share DNA with another section or share DNA, with another organism. They can share DNA, which is essentially sexual reproduction. Or one of these can break off and go and form its own daughter cell and become a whole other plant on its own. Yeah, if you think of it sometimes like plants, that helps my mind versus thinking of them like animals, even though, they're not really considered plants, but
They're their own thing.
yeah. Mhm.
I think this was mentioned earlier, but how much do we know about meiosis at this point?
Nothing at this point. We're gonna go over it today.
Okay.
Yeah. Okay, awesome. Yeah, so we'll do this section today.
And one more time, what were you saying about the origin of replication minutes ago?
So the origin of replication, so if you look at a 4 chromosome organism, then they could have 20 origins of replication. They can have a bunch. So what happens is at the origin of replication, it'll supercoil and form a bubble. Wherever that origin of replication is, will form an individual bubble. And then it'll just replicate. So if you zoom in at a chromosome, remember, let me draw it double stranded so that it's not confusing. So you have one chromosome, and it can have a data box here, which is just an origin of replication. It's a bunch of A's and T's together, because remember the A's and T's only have two bonds together. Yeah, I kept saying binds. Two bonds together, so they can break apart easier. So it might have multiple origins of replication, but then what will happen is it'll form a bubble here, and a bubble here, and this bubble will form here. Sorry, it's not perfect here. Something like that, where there's two bubbles, and then it'll work in the 5' to 3' direction, 5' to 3' direction,
Towards each
until
other.
it falls off. Yeah.
So what falls in?
Until the polymerases are done, until it gets to the telomeres at the ends.
Okay.
Yeah. Just that last term that I used was heterochromatin versus euchromatin. So heterochromatin is super coiled and tight, 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 and euchromatin is open and ready to let in polymerases.
So those are euchromatin bubbles
Uh-huh.
that have the TA boxes.
Or if it's ready to transcribe and translate as well, it'll open up
So they open up for replication
for a
purpose?
purpose, replication or transcription or translation.
And then do they close back up?
And they
after?
close back up. And some, even though they'll replicate and they'll open up for replication, for the remainder of that cell's life, it'll stay closed in the area that codes for the genes for a different cell. So like, for example,
So like the inactivated genes?
inactivated genes, yeah,
So the
will
open-
always be heterochromatin unless it's replicating. Am I
Okay.
saying that? Am I saying that in a weird way?
I think so. So like your kidney cells would be, what, heterochromatin is the
Yep.
closed one
Uh-huh.
in like your fingers, for example?
Yes,
Okay.
exactly.
And then the euchromatin in your fingers is the part that has to do with the fingers?
Yes, that will be open and ready for transcription translation.
So does the polymerase just not read the heterochromatin?
Yeah, it can't access, it can't get in
Access
there.
it.
Yeah,
Is it a similar block to the,
like the repressor.
yeah, right.
Well, so actually, just within the last, I think it was like 10, 15 years ago, they figured out that it's not always repressors, that a lot of times it's not actually controlled by repressors, that it's because it's so super coiled, they can't get in there. So repressors, they've just figured out, like, I mean, not recently, but semi-recently, that repressors are mostly used to turn up or turn down or turn off specifically, if, you know, because you still need it available.
Right.
But it needs to be timed or it needs to be organized, you know, so it's less like blocked forever.
Yeah.
Yeah.
It's just so cool to learn like the most fundamental thing about us ever. It's like 2 years ago that they changed this thing about it.
It's crazy the stuff we still don't know. Like that's what they're still trying to figure out is like the junk DNA. When I was in undergrad, they were like, oh no, that's just junk. And now they're like, no, it's not. So that was, I mean, in the last 20 years, it's crazy. Yeah.
So when it's looser, is it not wound around the histones at that point?
Yeah, it'll unwind around it'll unwind from the histones.
Okay.
Yeah, yeah.
So that's what you mean by loose.
Yeah. Let me show you different colors here. So does that all make sense? Maybe, so what you guys can do is, we only have a few more minutes, but maybe you can yourselves just practice what the organelles are. And what their functions are. Zoom in. Here's look like kind of little honeycomb things. Forget. Did I miss anything? So maybe try and see if you can recognize structures or draw it yourself, label structure and function. We have a couple more minutes. always struggle with where to start. Should we talk about the stem cells? Should we not talk about the stem cells? Talk about the stem cells?
Can I ask about your picture?
Yeah.
Are you, is that little box supposed to be a protein, or is it supposed to be, the cell wall? Or not
This
cell wall,
one?
but the little square?
These?
No, along the, is it,
Oh, this?
Yeah, yeah.
Zoom in
okay, so zoom
about what
in.
these are, yeah,
Okay, so that's our,
yeah.
What is the little permanent sign?
This one is cytoskeleton. Yeah, that's just the way I always draw it, but maybe my drawings don't help. You guys don't help me
No, that
when
makes
you see it, but that's just always how I draw.
We got.
Yeah, put that together.
So the nucleus is the eggs. The nucleus is the rough. That's where the monkey is from little ribbons. So then what's that? Oh, it's just
Okay, how's it going? Everybody feeling good? Ready to go? Yeah? Okay. You look a little especially quiet today. We're gonna be spinning. Okay, so what we're gonna start off with doing, so we're going to